This is the first part of your cell transport and cell membrane notes. So the first part of the cell membrane that we're going to talk about is its structure. And on the next slide, I'm going to go ahead and draw this so that you have a chance to draw it as well. But here's just kind of all the main points. The cell membrane has two layers, so it's called a bilayer. Remember, bi means two. So two layers of phospholipids. Phospholipids. Well, we know that a lipid is basically fatty acids. That's the monomer, right? But we're adding something to it. We're adding a phosphate group, and I'll show you what that is in a second. So we have a bilayer of phospholipids. That's most of the cell membrane. And remember, the lipids are made in the smooth ER. Proteins are also going to be in this membrane. It's not all phospholipids, and I'll walk you through some examples of proteins. Sometimes they function as transport channels. Sometimes they serve as markers, like a name tag. It tells the rest of the body what kind of cell it is. Sometimes they serve as like hormone receptors, and their job is to receive a signal. But there are proteins all over this membrane. Cholesterol, which is a lipid steroid, is also in the cell membrane, and that helps to keep the membrane fluid. Keep membrane fluid, which just means because it's a little bit of a bulky molecule, it's going to prevent it from getting stiff. I'll say no stiff, just to abbreviate. So it's going to keep things moving, it's going to allow things to move through a little bit, and it's going to prevent really these lipids from getting all tangled up in each other. All right, as a whole, the cell membrane controls what enters and leaves the cell. It's kind of like a boundary between the inside of the cell and the outside. It separates the internal cellular chemistry from the external environment, and that's going to help the cell maintain all sorts of homeostasis. Remember, that's just a balance of something needed for survival. It could be a balance of salt, sugar, pH, whatever the cell needs to keep the same inside as opposed to the outside environment. And an overarching model, you might have heard this in middle school, for the cell membrane is called the fluid mosaic model. And you'll see this better in a couple slides. But fluid means what you think it does. It means being able to move. And mosaic is the art term. So if you are an artist or you take an art class and you make a mosaic, you have like a whole bunch of different components, maybe that are different shapes or different colors, and you've arranged them into a mosaic pattern. Because there are so many things in the cell membrane, including phospholipids, including proteins, including cholesterol, right? These are kind of like little bits of pieces of a mosaic artwork. So we call it the fluid mosaic model. So I'm going to go ahead and draw just a little bit of membrane here. So most of the membrane is made of phospholipid. So this entire molecule is a phospholipid. Phospholipid. And there are two components. You have the two fatty acids. Those are the lipid component. And we're familiar with what lipids are. And then this ball, it kind of looks like a ghost. This ball on top is the phosphate group. Phosphate group. And if you're curious as to what that is, it's a phosphate, 4 oxygens, and it has a negative charge on it. But you don't have to know that. But if you're curious, there it is. And the entire membrane is made of these structures. So remember, it's a bilayer. So what you have on one side, you're going to have on the other side. So here's all your phosphates and your fatty acids. so all your phospholipids, and this goes all the way around the cell. And then on the other side, so maybe this is the outside of cell, then you have another layer with your fatty acids. Oops, those didn't quite line up. There we go. Inside. I don't know why I have the capital I there all of a sudden. Inside. So this is where the cytoplasm is. And remember that the cytoplasm is mostly water. And water is polar. Remember that from Unit 2. Water is polar. Oops. It has a partially positive side and a partially negative side. The same is true for the outside of the cell. The outside of the cell is mostly water, which means the outside of the cell is mostly polar. And the reason we're talking about this is because if you drop phospholipids in water, like a whole bunch of phospholipids like in water, they will spontaneously form a bilayer structure. You don't need any higher level orchestration or energy to do this. They will spontaneously form this bilayer. And the reason they do that is because of the chemistry of these molecules. So remember that anything that's polar is going to attract to anything that's also polar or that has a charge. Well, this big hunky phosphate group on top of this phospholipid has a negative charge. It has a full negative charge. So it's going to be attracted to some of the hydrogens in water. Remember, the oxygen in water is slightly negative and the hydrogens are slightly positive. Right? So this negative charge is going to want to stick to these hydrogens. It's going to want to make hydrogen bonds with them, which means that if you have an external watery environment and an internal watery cytoplasm, these charged phosphate groups will actually spontaneously align themselves with the water. So they are going to face the outside of the cell, and they are going to face the inside of the cell. And the term for this, the fact that they like to face the direction that water is called hydrophilic. They are water loving. They are hydrophilic, so they face the inside and the outside of the cell. The inner portion, the fatty acid portion, remember, is lipid. Lipids don't mix well in water. If you drop oil in water, just on everyday experience, they don't mix. And you can try it at home whenever you want. Just get some like olive oil or canola oil or veggie oil or whatever and drop it in water, and you'll see that it floats and it makes these little bubbles, and they don't really mix well with water. This is the same lipid. These are lipids, right? So they're not really going to want to mix with water at all. And in fact, they're going to hide from water. And they're going to hide from water in this middle portion, the interior of this bilayer. So this is where all the lipids are. We call this behavior hydrophobic, water fearing. So these lipids are hydrophobic, so they're going to hide in the middle of this membrane where there is no water. Whereas the phospholipids, because they have a charge on them, they don't mind water at all. They love other things that are polar and charged, so they're going to face the external environment. All right. So let's go ahead and add that. So we'll say that the phospholipids are hydrophilic and polar, whereas the fatty acids are hydrophobic and they're actually non-polar. They do not have a charge. And this is why you get the phospholipid bilayer, and it's incredibly important to understand these vocab terms, to understand what hydrophilic and nonpolar are, what hydrophobic and nonpolar are, what hydrophilic and polar are, and you should be able to explain why the membrane looks the way it does. So remember we talked about this being a mosaic. There are other things besides phospholipids. There are proteins and cholesterols, etc., right? So this is kind of a more complete visual. The golden components, these yellowy golden components, are the phospholipids. Note the phosphate group, right? These are the little circles that we just drew. And notice the two fatty acid tails coming off of each of them, right? And most of the membrane is made of this, right? The external environment, kind of up here towards the top, it says extracellular fluid, the outside, is watery, right? So you have all of the phosphate groups facing it. The inside, the cytoplasmic side, which is also watery, is also going to have these phosphate groups facing it because they are hydrophilic and they are polar. The middle of the membrane, where all of the fatty acids are, is hydrophobic and nonpolar. And there's no water in there, so all the little fatty acids hide in there because they're all scared of the water because they're nonpolar. All right. These blue things are proteins. So I'm just showing you that embedded within this bilayer are other things, right? So you've got protein markers or a recognition protein. Some proteins are going to have a little spot for things to bind to them because they might actually be enzymes or receptors. So they have a spot for a substrate to attach. Some proteins, like this protein on the right, have a channel through them, have a little gap through them so that molecules can move in and out. Sometimes these proteins have little sugars on them. So what you're going to notice is that over here on the left, this protein in particular, this recognition protein, has a carbohydrate on it, this big hunky carbohydrate on it. For cells, that carbohydrate serves as a name tag. It's like a name tag that says, hello, I'm a skin cell. Please don't attack me with your immune system. The reason that you have blood types like A, B, O, is because you all have different carbohydrates on the proteins that are in the membrane of your blood cells, right? So these serve as recognition markers and it lets the body know that those cells belong to you and that they're not foreign or anything like that. Here's the cholesterol we talked about earlier. So remember, the cholesterol is a lipid. It is a lipid, so it is hydrophobic, and it will stay in the middle portion of the membrane, right? And notice how big and bulky this molecule is. It's making gaps in the membrane here. So that's going to allow things to kind of move through and keep it from getting all stiff and rigid. Let's see, what else is on here? The glycoprotein up here in the top right is the same as this protein down here in the left. So you have kind of a protein with a glycogen or a bunch of sugars on it, right? So the glyco means sugar, and the protein obviously means protein. So it literally tells you exactly what it is. It's a protein with sugar on it. And again, that can like serve as a name tag. On the inside of the cell, all of this spaghetti-looking stuff is actually cytoskeleton. Remember, the skeleton in your body is attached to muscle and it helps you move. It's like a point of force where you can like apply pulling forces using tendons and ligaments and muscles, and you need that to help you move. It's the same thing for cells. If a cell needs to move, it has to provide a force on its membrane to kind of push off from. And also a lot of the structures in the cell membrane are attached to parts that are inside the cell. So the cytoskeleton all helps with that, right? So that's the cell membrane. All of these pieces can move. They're not stuck there permanently. They do kind of float around within the membrane. So that's why this is called a fluid model. And notice just all the different components, all the different pieces. That's why it's called a mosaic. So the fluid mosaic model of the cell membrane. How do substances move in and out of the cell? Because the cell membrane does regulate what comes in and out, so how does that happen? There are two things to keep in mind here. If you are a very small and non-polar molecule, you very easily can pass directly through the phospholipids. So if you have phospholipids, and you're a small non-polar molecule, let's say your oxygen, you can fit right through the membrane all on your own. The one exception to this rule is water. Water is so small that it actually can still fit, even though it's polar. So that is the one exception. If you're a larger molecule and or a polar molecule like glucose or amino acids, or pretty much anything other than some dissolved gases, you're usually way too big and too polar to pass through the membrane. The hydrophobic center of the membrane will reject you, so you need some help. So in that case, you're going to pass through what are called protein gates or protein channels, and I'll show you that now. All right. And you are responsible for knowing the characteristics of things that move through the membrane. So you should know that small nonpolar molecules can go directly through this bilayer, whereas larger polar molecules are too big and too polar and they need help. You do need to know that. Okay. So here's some more specifics. There are two types of movement, or two types of transport. We're going to look at these in turn. Passive transport is first. This is where molecules do not need energy. Do not need energy to move across the cell membrane. Active transport, you do need it. You do need energy. So molecules need energy. So let's see what each of these is like. Let's look at passive transport first. Molecules move with the flow from an area of high concentration to an area of low concentration. From an area of high concentration to an area of low concentration. Molecules do not need energy. Remember, this is passive. No energy needed to move across the cell membrane. There are two types of passive transport called diffusion and osmosis. So let's look at an example. All right, diffusion, again, this is passive transport, so you're going from a higher to a lower concentration. So let's draw this. So here's some space. Let's say you sprayed perfume. So there's my perfume bottle, and I spray it. And as soon as I spray the perfume, there's a really, really high concentration of molecules, right? Over here, let's say I'm on the opposite end of the room, there's no perfume molecules, right? So here, right next to the bottle, you have a high concentration. You have all of these perfume molecules packed together. And then on the other side of the room, where there aren't any, there's a low concentration of perfume molecules. So basically, all diffusion is saying is that because these molecules have kinetic energy, they are moving, right? They have kinetic energy. They are bouncing around and they're hitting each other. And when they hit each other, they bounce further apart. So as these molecules move, again, anything that has heat has energy, which means it has movement. And these are gas molecules, so they're not all stuck together like a solid. They're going to bounce around and they're going to start to disperse, right? So over time, these molecules, as they collide with each other and other gases, will get further and further apart. And this is why if someone sprays perfume in one corner of a room, eventually it diffuses, it spreads to the other part of the room and you'll smell it all the way on the other side. It might take some time, And the scent might not be as potent or as strong because by the time the molecules reach your nose all the way on the other side of the room, they're so far apart. But eventually they will diffuse, again, from a higher concentration right outside the bottle to a lower concentration everywhere else in the room. The same thing is, you can see this if you take, and I would actually encourage you to do this if you can, if you take a glass of water and you drop a food coloring in it, what you're going to notice is that there's a really, really high concentration of food coloring where you put it in the water. But because all of those food colored molecules are bouncing into each other and knocking into each other and repelling each other and moving all these different directions, eventually over time, the food color disperses to fill the entire container. Right, so eventually your entire container will be whatever color of the dye was, right? And again, this is all simply because of random kinetic energy. That's all diffusion is. It's just movement. It's kinetic energy, the energy of movement. And that's key. This takes absolutely no energy. No energy is required. This happens spontaneously. And you go from a high to a low concentration. Eventually, molecules will reach equilibrium. Molecules will reach equilibrium. So in our perfume example, there will be a time where in the room, the distance between all the perfume molecules is the same. So if this is your room, down here in the bottom right, they're kind of uniformly distributed throughout the room. And it smells equally of perfume everywhere, rather than super, super smelly in one corner and not smelly at all in the other corner, right? So we say when this has happened, when everything is evenly distributed, the molecules have reached equilibrium. If you have to go through a membrane, you can diffuse right on through it if you're small and nonpolar. But if you're big, so let's say that we're dealing with glucose. Let's say this is glucose. Glucose not only is big, but it's also polar. It can't fit. It won't go through this bilayer. It's too big, and the hydrophobic fatty acid tails won't let it. So it needs help. It's going to have to go through a protein channel. This is called facilitated diffusion. You're still diffusing. Notice that there's still a high concentration of glucose on one side and a low concentration on the other. So we're still diffusing, but because there's a membrane in the way, right? We're not just going through the air right now. Now we have to go through a membrane. We need some help to do this because we're big polar glucose, right? So we need to go through a doorway. That's what this protein channel is, through a transport protein. Protein channel or transport protein. These are synonyms. They mean the same thing. All right. Just a little aside is that the channel that the glucose is moving through is unique to glucose. Unique to glucose. So let's say that I had an amino acid. I'll make it a square. So here's an amino acid. The amino acid is also too big and too polar to move through the membrane. But because this channel protein is unique to glucose, it wouldn't be able to go through. It would have to have its own channel unique to itself. A little drawing here, a little tunnel through the middle of it. It would have to have its own channel to move through. It wouldn't be able to share another molecule's channel. All right. If water is the thing diffusing, it gets a special name. It's called osmosis. Again, this is still passive transport. There's still no energy required, and this is still diffusion. But because water is so important in biology and everywhere, it gets its own special name. So osmosis is the diffusion the movement or the diffusion of water through a membrane from an area of high to low water concentration. Nothing new. The only difference is now the molecule. It's not a perfume molecule. It's not glucose or anything like that. It's a water molecule. So it gets its own special name called osmosis. Right? And again, water is always going to be moving, even when it reaches equilibrium. So here are two cells, and they are separated, well, in this case, by two bilayers, because each cell has its own membrane. But what you need to figure out when you're looking at cells like this is, where do you have a higher concentration of water? Right? Water will move from the cell with a higher concentration to the cell with a lower concentration. So which cell is higher concentration and which is lower? Right? Well, the cell, okay, we'll label this cell A and this cell B. Let's start with cell B. Cell B has quite a lot of salts. This is some kind of solute. This could be salt, it could be glucose, it's something in the water, right? And this takes up space, right? Salt and glucose and amino acids and dissolved particles, they all take up space, right? Which means that's less space that water can take up. Whereas A, cell A, has fewer salts. Right? Here there's only like 6, and over here there's like 15 or something, right? Which means because there's so less salt, there's so much more water. So cell A has more water. It has a high concentration of water. Cell B has a lower concentration of water. And because water will diffuse across the membrane, it will move from high to low concentration from cell A to cell B. And that's what these water arrows here are showing you. Water's moving from cell A to cell B, from a high water concentration to a low water concentration. And yes, that means cell B is going to swell, and it could actually pop, whereas cell A is actually going to end up shriveling a little bit. All right, the last kind of transport for this video is active transport. Active transport requires energy. Remember that cellular energy is in the form of ATP. So whenever you see ATP, I want you to remember that that's just energy. Right? That's just energy. And this is the opposite in terms of movement. So we're moving from a low concentration to a high concentration. So this is like going against the flow. Think about like when you stuff a suitcase. Stuff a suitcase. All the stuff is on your bed or on the floor or somewhere else, and it's kind of all, it's not all packed together, but you have to make it all fit in a tiny space. So what do? You open the suitcase and you cram it all in there, right? You stuff it all in there. You're taking it from a low concentration of clothes in your room and you're stuffing it into a high concentration of clothes in a small suitcase, right? And anyone who's stuffed a suitcase knows that the more and more stuff you put in there, the harder and harder it is to close. And you can struggle with it. You can try to get the zipper closed. Sometimes the zipper gets caught. Sometimes you have to sit on the suitcase to make the top close. But the point is, it takes a lot of energy to stuff a suitcase, right? If you want to cram a bunch of stuff in there from a low to a high concentration, you need to provide a lot of energy. And it can be quite a little workout to stuff a suitcase full of a lot of things. And this is why it requires energy to do active transport. These are molecules that want to naturally diffuse, but you're stuffing them together against their will, basically. And that's why this requires energy. And there are three main ways this happens. So let's look at each of these in turn. The first is a protein pump. A protein pump. And while I talk, this little video is going to play. But again, this is active transport, so you need energy. And proteins are going to move large molecules across the membrane from low to high concentration. So let's take a look and see what happens. Okay, here's the start. So you have sodium. You've got 3 sodiums entering this pump with the help of energy. This little blue molecule is energy. And then you get forced to the outside of the membrane. The potassiums, the Ks, do the same thing. And they get forced to the inside of the membrane. Watch it again. The 3 sodiums, the green sodiums, are coming in to the protein pump. The blue energy molecule, the ATP, gets attached, and they're pumped to the outside of the cell. The potassiums, the Ks, do the opposite. They're getting pumped, using energy again, to the inside of the cell. Notice that these are moving from a low to a high concentration. Right, there's a high concentration of sodium outside the cell. Potassium is now getting pumped inside the cell to where it has a high concentration. Right, this is the opposite of diffusion. And you need energy. You need this little blue ATP molecule to help you do that. And you need proteins. You need protein pumps to do this. Let's look at active transport's last 2 examples, and then we'll wrap up. Endo and exocytosis, right? So endocytosis first. I swapped these, that's okay. When a cell takes in material by making pockets in the cell membrane, those pockets then become vacuoles and potentially lysosomes if you're in an animal cell. Exocytosis is the reverse. Cells send materials out. A vacuole fuses with the membrane and releases the content. And again, both of these require energy, and they are both active transport. This is bulk transport. All right, this is where you go to either take things in or ship things out en masse. You have a lot of proteins or a lot of amino acids or whatever it is. So here's the picture. On the left side, we're showing you endocytosis. So there's a smaller cell or a virus or a protein coming in. There's something coming into the cell that is simply too big. It's too big to fit through the membrane. It can't do that. Nor can it fit through a protein channel because it's still too big. So this is for really, really big molecules or small organisms or viruses, right? The entire membrane pinches around it and encloses around it, pinches off into a vesicle that can then become a vacuole, or it can become a lysosome or a peroxisome, and it can get digested and get broken down, right? This is endocytosis. Endo means into, coming into the cell. Exo is the opposite. So here, maybe you have a whole bunch of proteins that you just packaged at the Golgi body. And you're going to send all of those proteins outside the cell. So this, let's say that this vesicle just came off the Golgi. It's going to make its way over to the cell membrane. It's going to fuse with the cell membrane and release the contents. All right, this is exocytosis. Both of these require ATP. So the last thing that I want to talk about before I end these notes, I'm actually going to go back. I'm going to go all the way back to our drawing of this cell membrane, because I forgot to tell you this. We call this a semi-permeable membrane. A semi-permeable or semi-permeable membrane. Semi means some or part or half. Permeable means things can go through it. So what things can go through and what things can't? Right? Well, our small nonpolar molecules can go through it. They are permeable to the membrane, or the membrane is permeable to them. And the large polar molecules cannot. So this is why the membrane is semi-permeable. Some things, the small nonpolar things, and water, remember water is the exception, can go through, whereas larger polar things cannot. All right, so if you ever see the word semi-permeable describing a cell membrane, that's why. And if you go back to our example here, where we talked about osmosis, this is also why water moved, but the salt did not. Notice the salt did not move. The salt stayed in the cells. It didn't move to the other cell. And that's because salt is a large polar molecule, right? Whereas the water, even though it's polar, is small enough to fit through the membrane. So this is an example of the semi-permeable nature of the cell membrane. Small molecules like water are able to fit, whereas the larger polar salts are stuck on their side of the membrane. I hope this helps, and we will talk about this more in class. See you later.